Welcome back. So I just wanted to reiterate to you the fact that learning takes a lot of time. So make sure you space your learning over time. Be repetitive about it. Make sure you keep up with your reading, you keep up with your study guides, keep up with your root words, and do all the things you need to do to be a great anatomy student. Well, in this lecture, we're going to talk about histology. Histology is the study of the tissues of the body. So this is going to be something probably you've never had before. So I hope you'll enjoy learning about the tissues. We'll spend a lot of time in lab studying tissues and getting to know what they look like under the microscope. Just to refresh your memory, tissues are made of cells, and so cells have certain parts that allow them to function. We've talked about the nucleus before. The nucleus is that part that stores the genetic material and allows for instructions to pass out so that you can build proteins. Of course, the endoplasmic reticulum is the part that's going to build proteins, and then those proteins that it builds is going to be sent to the Golgi apparatus to be packaged into little vesicles, which eventually might become lysosomes, or they might be exported out of the cell. We do have another kind of endoplasmic reticulum, smooth endoplasmic reticulum. It does things like synthesize lipids, it processes sugars or carbohydrates, it makes more phospholipids, so it makes more cell membrane, and it can detoxify drugs and alcohol. In some cells, like muscle cells, it stores calcium. So we have talked about the flagellum. That's a structure that allows sperm cells to move. Lysosomes are little packages of digestive enzymes that help to digest food. Peroxisomes are another structure that helps us to break down fatty acids, and it also can detoxify drugs and alcohol. And cytoskeleton, is kind of the framework. It's the bones and muscles of the cell to give it a framework for allowing it to change its shape and even to maintain integrity. Every one of the cell parts you see there is held in place because of the cytoskeleton. We can never forget the mitochondrion. This is the structure that's going to form ATP. It's going to take your food, and it's going to take the oxygen that we breathe, and it's going to make ATP, which is the energy that's going to run the cell. And of course, one of the most important structures to create a boundary is the cell membrane or plasma membrane. So that's just a quick review of some of the cell parts, because tissues are going to be made of lots and lots of cells. You always need to know the cells and how they function in order to understand a little bit better how tissues will be formed. So just to reiterate the fact that the plasma membrane is really important, it's a phospholipid bilayer, but it's a mosaic of parts. So you have proteins that go all the way through the membrane. Those are those integral proteins. You have proteins that can sit on the outside surface or inside surface, and those are called peripheral proteins. We do have cholesterol, which helps the cell membrane not get too fluidy or too solid, depending upon the temperature. And then we have the glycocalyx, which makes up the extracellular matrix. And this is made of all different kinds of glycolipids, glycoproteins, and other things we'll talk about later on. On the inside of the cell membrane, we do have the cytoskeleton many times attached to these peripheral proteins. That allows for the cell membrane to change shape if it needs to. Okay, so what exactly is a tissue? So by definition, a tissue is just a collection of similar cells that work together to perform a specific function. Now, how many cell types do you think the human body has? Well, scientists are actually studying that right now. There's a human cell atlas that's being formed, and we don't actually know how many different types of cells there are yet. But there are many different types of cells. We'll be talking about some of these cells through the course of this class. So if these cells come together, they form a specific function. We call that a tissue. So cells, when they come together, are forming a new emergent property. The cells together have a different job forming that tissue than they had as individual cells. So that's some of the complexity that we're beginning to form as we keep on adding new levels of understanding of the human body. So how is a tissue made? Well, a tissue is made by having cells come together. Okay, so let's see how they do that. So we do have many different kinds of cell junctions. These are intersections of where cells come together, and they can come together in many different kinds of ways, and there's many different kinds of attachment points. So in this particular lecture, we're going to talk about gap junctions. We're going to talk about tight junctions, adhere injunctions. We're going to talk about desmosomes and hemidesmosomes. And what we'll do is we'll look and see how each of these junctions does a particular job to allow us to form tissues and then to allow the tissues to actually work in a desired way. Let's start with tight junctions first. So tight junctions, as you can see, are tight junctions. They're where cells come together. So this would be cell A over here and cell B. It's where cells come together, and they're so tightly joined together by these strands of proteins that you can see here and here. They're so tightly joined together that things can't come in between them. Okay, so in this intercellular space, you could have stuff out here, but you see that stuff can't pass down through these different layers. So you can probably see why these would be important. Like in your intestines, you don't want bacteria out here entering in through these little spaces in between cells and then entering into your body and begin to eat you as food. Okay, so in your urinary bladder, you definitely don't want urine going through the urinary bladder and leaking into your pelvic cavity. So we have these tight junctions to create barriers so that it actually forms a boundary in between these cells that are joining together. Tight junctions are really super important in various organs of the body. So adherent junctions form like belts of adhesion. So generally speaking, what you have on cell A is you have on the inside of cell A, you have this plaque, which is this proteinaceous substance that has these smaller transmembrane proteins that are stuck to the plaque. And on the other side in cell B, you can see that we have a plaque, and then we have also the little glycoproteins that are able to almost like zipper and attach. You can see it almost like zippered together, and are attaching cell A and cell B together. And these are sheets, so they go around the whole cell. And if you went back to the first diagram that I showed you in this, you would see that it's a whole sheet of connectivity. And what this does is allow cells to connect to each other and provide some flexibility so that they can remain attached together, but yet give some flexibility to the cells as they're moving. So as food's passing through your intestine, you want the cells in the intestine to be able to have some flexibility and expand some, but you don't want them to fully separate. And this is what an adherence junction is actually doing. This is found in other places in the body though, but it's very easily seen in the digestive tract, the esophagus, small intestine, because food will pass through those things and cause some distension to occur, and you want to have some flexibility in those tissues. Well, desmosomes are going to be almost like spot welds. Some people call them cell rivets, and they have a structure that's similar to the adherents in that they have a proteinaceous plaque. So it's just a collection of proteins here, and that collection of proteins is anchored in by these parts of the cytoskeleton. Embedded in the plaque is another one of those little glycoproteins, and you can see they're zippered together in order to give us a nice connection there. So you can think of that as like a spot weld, and this is going to be found in tissues as varied as tissues of your skin and your heart muscle as well. So again, it gives some flexibility, but yet it is strong and doesn't allow the tissues to tear apart. Now, hemidesmosomes are going to be, you can see hemi kind of means half. So you do have your plaque, you have your protein anchor, you have also a glycoprotein down here that's going to anchor this plaque into what we call the basement membrane. Now, this is a very specific kind of attachment. It's an anchoring point into a basement membrane. A basement membrane we'll talk about in just a minute, but this is an acellular proteinaceous layer that the cell above secretes below, and it serves as an anchor point for the attachment of these cells, so that they can anchor to a place, they won't be floating around, and it also serves as a place that they can wander around and move across. So this is a hemidesmosome. It's kind of a more specific kind of cell junction. Specifically, we're going to find this in what we call epithelial tissue, and so you would find this in, say, your skin cells. Gap junctions are very specialized, and they're composed of these proteins called connexins that are forming little pores that molecules can move into and out of, cell B and cell A. Okay, so we can have molecules over here, they can move through those pores and go over to B, and vice versa, they can go from B over to A. And we find these very specifically in embryonic cells, so you're going to be more likely to find these in embryonic cells, and then cardiac muscle tissue, that is muscle tissue found in the heart, is going to use these particular junctions as well. And what these things do is they allow for chemical communication between adjacent cells. And in embryos, chemical communication is really important so cells can form tissues and form organs in the right location at the right time at the right place. Cardiac muscle cells actually transmit electrical signals through these pores so that the heart muscle will contract in the right way. So that's just two different examples of how gap junctions are useful to cells. Okay, so and you can see just here, you can see the tight junctions, not allowing bacteria or chemicals to leak in or out of the cells or in between the cells. You can see the adherence junctions, where it forms like a sheet-like protection and connects to cells in all three dimensions around this one particular cell. We talked about the desmosome, and that's the cell rivet or the spot weld, and that helps to give strength to a cell. We talked about gap junctions, which allow chemical communication between cells, and we talked about the hemidesmosome that allows for connection into the basement membrane. Okay, so that's how tissues are going to be formed, and they're formed because of the connections they have between adjacent cells. Now, we're going to talk about 4 types of tissue, and you might think, well, that's great, we only have 4 types of tissue, but each type of tissue is going to have sub-levels to it. So there are many different kinds of epithelial tissues and connective tissues, only 3 muscle tissues, and we're only going to talk about 1 nervous tissue today. So the 4 types of tissues include the epithelial tissue, and this would be tissue that's found like in your skin. If you take your tongue and run it around inside your mouth, that's all epithelial tissue that your tongue is feeling, and it's covering the surface of your tongue. There are 3 types of muscle tissue. We're going to talk about skeletal, cardiac, and smooth muscle tissue. There are many types of connective tissue: bone, blood, loose connective tissue, and so we'll talk about those, and then we'll talk generally about nervous tissue. We have we really have a whole chapter on nerve tissue, and we really have a whole chapter on muscle tissue. So, ones we'll focus on mainly today are going to be the epithelials and the connectives, but we'll just briefly talk about muscle and nervous as well. This just is a graphic showing you just generally some of the places where you can find these different tissues. Nerve tissues you can find in the brain, spinal cord, and of course your nerves. Muscle tissue you can find in your skeletal muscles that attach to your bones, your heart, and then the walls of your organs like your esophagus, stomach, small intestine, large intestine. Epithelial tissues are going to be found just about everywhere. They are all over the surface of your skin. They line all of your digestive organs from your mouth all the way to your anus. So they're found in many different locations. And connective tissues are found everywhere as well. Bones, tendons, ligaments, fat, blood, all of that's going to be connective tissue. So what I want to do is I want to start with the epithelial tissues, and I want to kind of teach you about these tissues and prepare you for when we see that in a lab. So epithelial tissues are going to have certain characteristics that they have in common with other epithelial tissues. These are very specific characteristics that these specific tissues actually contain. So one thing that we see in epithelial tissues is that they have polarity. That is, they have very defined specific sides to them. So, for example, there is an apical or free surface, and this typically could be smooth. It could have microvilli. If you remember, microvilli are these little extensions of the cell membrane. Or this apical surface could have cilia. So if you looked at the cell, it would have little cilia on the surface that are going to create waves of movement. So we do have a free surface. This is a surface that's not attached to another cell, but typically it extends to the to a lumen of a tube, to the inner surface of a tube. It could, so like if you looked at say the intestine, the cells, this is where food would be over here. So the cells, this would be the apical surface making up the wall of that tube. This would be the apical surface at the top of it. So that's where you would find it being smooth, or microvilli, or having cilia. Okay, so next we have the basal surface. This is the surface where the epithelial tissue is attached. So if I again made a tube, let me go back and make a tube. If I made a tube, and you can see this would be the apical surface and this would be the basal surface. Okay, so they have polarity to them, an apical and basal surface to them. The basal surface is going to sit on a little acellular proteinaceous membrane called the basement membrane, and we'll talk about that more in a couple minutes. Epithelial tissues by definition or by characteristic are going to not be vascular. Okay, so there's not going to be any vascular tissue that goes through them. And I'm sure you've given yourself a paper cut before and it didn't bleed, because what you did is you cut through the epithelial tissue, but since there's no blood vessels in there, it didn't bleed. So they are avascular. They don't have blood vessels. They do have nerve endings that extend into them, maybe even sensory nerve endings or pain nerve endings that extend down into them. So you can feel epithelial tissues and sense them, but they're not going to have blood exactly in them. So there's no blood vessels in them. Now, not having blood vessels inside of you means you have to be close to blood vessels, because you have to be close enough for gases like oxygen and carbon dioxide to diffuse into and out of your cells. Now, these epithelial tissues are going to be rapidly dividing, and they're very, very, very quick to divide. When you cut yourself, it takes just a very short period of time before that wound heals. Of course, if it didn't, you would leak blood out and die. But the epithelial tissues are very quick. So I'm sure you've had an ulcer in your mouth before, and you saw how quickly that healed. Within a week, it goes from having a hole all the way to being healed. So this is great that they do this, but it's also kind of bad that they do this. Rapidly dividing cells sometimes can get mutations and become cancerous. Okay, so if you think about the different kinds of cancers people get, lung cancer, well, that's an epithelial. Lung tissue is epithelial. Skin cancer, skin is epithelial. Stomach cancer, well, that's an epithelial tissue. Colon cancer, that's an epithelial tissue. Pancreatic cancer, that's an epithelial tissue. Now, I'm not saying every cancer you get comes from epithelial tissue, but the great majority of cancers that humans get, cervical cancer, vaginal cancer, all of these things are going to be epithelial. And you might ask yourself, well, why? Well, one reason that more cancers are more likely to come from epithelial tissue is because they rapidly divide. But another reason is that they're exposed to the environment. Okay, your skin's exposed to ultraviolet radiation. Your large intestine or small intestine, those are exposed to things that we eat in the environment. So they're more likely to be exposed to carcinogens that are in the environment. Another characteristic of epithelial tissue is that the cells are closely packed together. Okay, they're very tightly packed together to form that tissue, and that helps to form an organ that works correctly. Of course, they form sheets that are held together by the junctions we just talked about. They have great regenerative properties. They can regenerate themselves very quickly, and because they can divide very rapidly. And if you ever look at an epithelial tissue, here's the apical side, here's the basal side, here's the basement membrane. The epithelial tissue is always going to have connective tissue underneath. So this is where your connective tissue would bind the epithelial tissue to underlying layers such as muscle or the coverings of muscle. Okay, so they're supported by connective tissue. That connective tissue will have blood vessels in it. It'll have nerves. It'll also have anchoring points, so it can be bound and anchored to underlying tissues. Well, I think epithelial tissues are quite beautiful. I mean, if I just take a look at these tissues right here, you can see some of the basic parts of it. So we have here the apical surface. There are also lateral surfaces, how the cells connect or communicate with each other from the side view. There's also the basal surface. So we have polarity to these particular cells. So epithelium, this is just an epithelium here. And you can see the basement membrane down here. The basement membrane is made of a basal lamina and a reticular lamina. These are just proteinaceous layers. These are typically secreted by the cells that are surrounding it, and they serve as attachment points. So the epithelial cell is attached to the basement membrane. The connective tissue is attached, and so they're all connected together. Here down here we have the connective tissue, and you can see a blood vessel there. So you can see the epithelial tissue doesn't have blood vessels running through it, but they're close by so that gases can diffuse up and food can diffuse up and waste products can diffuse down through that. The basal lamina is adjacent to the basal polar surface of the epithelium, and it's a very thin supporting sheet. It's non-cellular and it's adhesive. It consists mainly of the glycoproteins, proteins that have carbohydrates attached to them, and these are secreted by the epithelial cells. This basal lamina also has collagen fibers. Collagen fibers have great strength to them and will allow for the cells that are attached to the basal lamina to be connected pretty tightly. So it is a selective filter, so there are molecules that can move through that proteinaceous layer from underlying tissues or from the epithelial cell to the underlying tissues. And it acts as a scaffolding, okay? So it acts as a scaffolding that epithelial cells sit on, so you actually can build a tissue by having the scaffolding. You know, one day when we have 3D printers that print tissues, they're going to have a basal lamina, and they'll actually take and begin to place epithelial cells on the surface of the scaffolding, and they'll grow, and they'll use the scaffolding to migrate and to form a layer of tissue. Our cells use the scaffolding to actually migrate, to move around, to repair a wound. Okay, so the basal lamina is really super important. 3D printing will one day create tissues. I mean, it's already doing it now, but they're just not using it for human use yet. But one day we'll be able to create tissues and organs using 3D printers. And because of our knowledge of the basal lamina and how these tissues are arranged, we can actually build these things instead of relying on people donating organs. So there are many functions of epithelial tissue. One of the main functions of epithelial tissue would be protection, so physical protection of your body. So you can think about the protection your skin awards you, the inner linings of your mouth, very, very protective. They also control permeability, so this is another function of it. There are all kinds of proteins that are on the surface of these tissues that allow certain substances into and out of the cell, so they do control permeability. One of the great places that you have great permeability would be in the kidney. So we have lots of epithelial tissues in our kidneys that allow for us to filter our blood out. Things that are toxins in our blood. There's a system there that is built of epithelial tissues that allow us to get rid of waste out of our body. Of course, epithelial tissues provide sensation. If you just take and run your finger over your arm, you can actually feel that because they are innervated. And this word right here means they have nerves going into it. They produce specialized secretions. So there is glandular epithelia that produces various things like breast milk or oils, like the oil secretions of your skin, or saliva. Okay, so there are a lot of different kinds of epithelial tissues, but we basically will name them based on how many layers of cells they have and what cell shapes the cells have. So you can have one layer of cells and be called simple. You can appear to have two layers, but really only be one layered, and that's called pseudo-stratified. Pseudo means false, stratified means layered. So you look like you're layered, but you're really not layered. because they basically have the nuclei are in different locations, which gives it an appearance of having multiple layers, but really they're all attached to the basement membrane. They're all one cell attached to the basement membrane. Or you can have many layers and be stratified. Okay, so that's based on arrangement of the layers of tissues. You can be simple, pseudostratified, or stratified. Now you can have different cell shapes. You can have a squamous or scale-like shape, and that's typically from a top view. So you can see it kind of looks like a scale from the top view. From a side view, though, you're going to be really super thin. Okay, so these cells are really super thin from a side view, but from a top view, it gives you almost like a scale-like effect. These cells will look like scales. So that's a squamous or scale-like shape. You can have a cuboidal shape. Cuboidal is a cube-like shape. That's not going to be a real perfect cube. You know, it might be kind of like a rounded cube, but it's kind of cube shape. And then you can have a column shape, and that's going to be what we call columnar. Now you can put all these things together to make different names. So for example, in this chart you can see there can be a simple squamous, so one layer of flat cells. You can have a simple cuboidal, which will be one layer of cube-shaped cells, or you could have a simple columnar, which would be one layer of column-shaped cells. But you could also have stratified squamous. This would almost look like a brick effect. Okay, so many layers of flattened cells. You can have a stratified cuboidal, which is typically, I think I've seen that in 2 layers. So you can have multiple layers of cube-shaped cells, and you finally can have 2 layers of column-shaped cells. So there's certain places in your body that have simple squamous. There's certain places in your body that has stratified columnar. Okay, and each of these different kinds of arrangements has a specific shape, and therefore it has a specific function. Okay, so where you have stratified things, you think of kind of like protection. Where you have simple thin things, you think of like diffusion of gases into and out of the tissues. Lastly, we have a transitional epithelium that doesn't fit any of the categories above, because the cells will change shape based on what the organ is doing. So one place you have transitional epithelium will be your urinary bladder, and when your urinary bladder is full, the cells look more squamous. When the urinary bladder is not full, the cells will look, you know, more column shaped or cuboidal shaped. So it changes shape based on the stresses that are applied to the tissue, and so therefore it doesn't fit any of the other categories. So there, you know, the types of epithelial tissue, we have epithelial tissue that serves as covering and lining epithelial. These typically will cover and line the internal and external surfaces. So they cover your organs and cover all the internal parts of your body, and they make up membranes for your cavities. We also have glandular epithelium, and these will be tissues that will be secretory. That is, they make and secrete products as part of glands. What I'd like to do now is take you through and talk about each of the different epithelial tissues and show you photo micrographs. Those are photographs taken under a microscope, and artist rendition pictures of this, so you can get a sense of it. You know, after doing lecture and lab and doing all this stuff, you should be able to identify the different kinds of tissues by looking at them through the microscope. So simple squamous epithelium consists of a single flattened layer of cells. So up here you see a side view or a profile view, not a top view. So you can see they're thin. They're sitting on a basement membrane. You can see the basement membrane right there. From a top view, they're going to be more scale-like in effect. Okay, but this is from a side view over here. This is a photomicrograph over here. You can see they have a pretty well-defined nucleus. You can see the cell sitting on the basement membrane. You can see the apical surface. You can see the basal surface and the lateral surface of these cells. So the reason these things are simple or only one layer is that you have substances that are going to diffuse or pass through this tissue really easily. So if you want oxygen to come through, you're going to have to be thin. If you want carbon dioxide to go through, you want to be thin, and nutrients too. A lot of times we find these simple squamous epithelial tissues in your lungs. That's evident. Also, they'll be found in blood and lymph vessels, because especially capillaries in the smallest lymph vessels, because you want fluids and gases and nutrients to move through them. And then some of your membranes that line body cavities are also going to be really super thin too, and they'll just be a thin boundary of simple squamous epithelium. This is just, I like this artist rendition because it shows you kind of a side view. This is the side view or profile view, and then it shows you a top view. So when you're looking at these in lab, you want to make sure you understand, am I looking at it from a top view or a side view? Because from a top view, you might be inclined to call it cuboidal, because maybe it almost looks cube shaped, but you have to be careful knowing that from a side view, though, it's going to be thin. Okay. You can see the basement membrane there. You can see the underlying connective tissues, and there's a blood vessel that would feed these tissues through diffusion. Okay, well, our next tissue is simple cuboidal tissue, and you can see it's one layer of cube-shaped cells. They typically will have a central round nucleus. They will be sitting on a basement membrane. A lot of times these simple cuboidal cells will actually form tubes. So what you're seeing up here in this photomicrograph is a tube. That's the hollow part of the tube, and that's the wall of the tube on the outside. Now the reason it's circular is because if you took a straw and cut it in cross section and looked at it from the front, so a straw, if you cut it in cross section and look at it from the top, it's going to be a circle. So what you're seeing in these pictures up here are lots of tubes that have been cut in such a way that you're seeing all the circles. Now sometimes you can get a cut of this where you'll actually see the wall of the tube and you'll see both sides of it. So it may look something like this. So this inner surface, this inner hollow surface would be that part right there. But if I cut through it, I can look at it like a tube, like a circle like that. So that's something when you get into tissues, thinking three-dimensionally will aid you in understanding how these things are arranged. Okay, a lot of people have difficulty seeing those tissues like that, kind of rotating them in their mind, trying to see them three-dimensionally. These function to form glands, so they function in secretion, making and secreting various kinds of products, and they also function in absorption, okay, in the kidneys. So in your kidneys, we're going to, take and filter blood out, but we don't want to get rid of all of the fluids that come into the kidneys because we would urinate all the water out of our body. So we have the ability to absorb some of the materials back into the body, and then some of it we send to the ureters, into the urinary bladder to make urine. So the simple cuboidal epithelium is also found in your salivary glands. It's found in your kidneys, your pancreas, and also the liver. So this is where you would find simple cuboidal epithelium. It's one of my favorite epithelial tissues to look at. It's a really interesting tissue. And again, I like this artist rendition because it gives you the three-dimensional aspects of it. So you can see what it looks like from the top view and from the side view. You can see the basement membrane, the connective tissue, and you can see it's always close to a blood supply. So you can have the movement of fluids into and out of the blood vessel into those tissues. Well, here we have a simple columnar, and this is one layer of elongated or column-shaped cells. A lot of times the nuclei are at the bottom near the basement membrane, as you can see in both the photomicrograph and the artist rendition. A lot of times the simple cuboidal, simple columnar epithelial tissue will also have mucus cells, okay, also known as goblet cells. And these goblet cells produce mucus and send the mucus out to the space here. And, the mucus in the large intestine is really good because it helps waste products slide out of the large intestine. Okay, so mucus being produced in your mouth and esophagus helps food to move down into the stomach. So sometimes you'll have those goblet cells there, and then you have, of course, your columnar cells. Now, the functions of these cells is to provide for movement of mucus, as we just described. Oftentimes, these simple columnar cells will also move in female fallopian tubes. They'll move the oocytes, the egg cells. They also serve as protection, and they serve as secretion of digestive fluids. In your small intestine, they absorb food, and in the small intestine, they're going to have microvilli that allow them to increase the surface area of the membrane and therefore bring food easily into the body. And those goblet cells will secrete mucus in many places, and that mucus is really important. It's a slippery mucopolysaccharide. It's a protein with carbohydrates. It's really slippery and slimy, and is really good for lubricating tubes that are in your body. So this looks like tissue we'd find in the small intestine. This is a non-ciliated simple columnar epithelium. Sometimes simple columnar epithelial will have cilia to help move that mucus. This particular tissue that the artist has done has microvilli. You can see the increase in surface area is tremendous. And you can see a mucus or a goblet cell. And these would be the simple columnar cells that are helping to absorb food. Definitely have our basement membrane there, and we have the connective tissue layer binding it underneath. That's a beautiful tissue to look at when you look at it under the microscope. This one is a ciliated simple columnar epithelium. So here we have cilia instead of microvilli. And remember, those beat in rhythmic waves of motion to aid in the movement of mucus. This is something you would see in the fallopian tube, or perhaps you'd see these in some of the respiratory tract airways, helping to move mucus around. Well, we have a pseudostratified columnar epithelium. These are going to have nuclei located in different positions, therefore it makes it look like it's stratified, but it's actually not. They contain, goblet cells, so you can see the goblet cells up here. They contain cilia. That's artist rendition. This is the photomicrograph. The cilia are beautiful here, and the goblet cells are really cool as well. This tissue will also line airways. It keeps your lungs clean. So if you smoke cigarettes, it actually destroys the cilia. And if you've ever seen somebody that smokes a lot, they have a cough. That cough is the smoker's cough, and it occurs because their cilia have been damaged or destroyed. And to get rid of the mucus that's in the airways, people have to actually cough it out. And here's what a pseudostratified ciliated columnar epithelium looks like in regards to artist rendition. You can see the cilia, you can see the nuclei are at various different locations, making it look like it's layered, but they are connected to the basement membrane and connective tissue underneath. And we also have transitional epithelium. This is a many-layered epithelium, and the cells will change shape based on the stress that you place upon them. Urinary bladder is a place that you can find this, and the urinary bladder can stretch and hold urine without leaking because these cells can change shape and yet maintain their integrity. And you can see there might be a tendency for you want to call that stratified cuboidal, but it does change shape. This is the relaxed view of it. But you can see in the relaxed view, and this is the unrelaxed view. In the unrelaxed view, it almost looks like the cells become stratified squamous. So we want to be very careful as to making sure we understand that's transitional and not cuboidal or a stratified squamous. Well, here's a stratified squamous epithelium. You can see it's many layers of thin cells or scale-like cells. And this occurs in your skin, the vaginal canal, the mouth, the throat, the anal canal. So it's found in places where you would expect a lot of friction to occur, a lot of protection that's needed, and that's exactly what it does for the human body. So it does form a protective layer. And so the skin on inside the mouth doesn't have a dead layer of skin cells. The skin that's over the surface of your body does have a dead layer of skin cells. So the stratified squamous epithelium does change in its shape depending upon what part of the body you're actually talking about. Okay, and that's artist rendition. So from the top view, you can see it does look scale-like. From the side view, it does look thin. It does look almost columnar / cuboidal at the very base of it. So when we talk about the integumentary system coming up, we'll talk about these cells having the function of reproducing very rapidly so that you get a progression of cells that moves this way, and then of course dead skin cells flake off. So there is a movement from the basement membrane to the surface and then off the surface. This is what skin looks like. It actually has a dead layer. It's a waterproofing layer of protein called keratin. And this is a beautiful photomicrograph showing you the layers of tissue, the layers of cells. And if I just went back there, of course we do have the connective tissue there, and the basement membrane is located right there. Well, here we have a stratified cuboidal. So it consists of several layers of cube-shaped cells. Typically it's 2. These oftentimes will form a tube. The lumen is the inside of the tube. And it forms a protective tube. And this is found in mammary glands. So milk production is going to be done in a stratified cuboidal epithelium. It's found in sweat glands, salivary glands, pancreatic glands. Also, you will find the the tube coming out of the urinary bladder that urine flows through to exit out of the body, the urethra, also has stratified cuboidal epithelium. So, protective tubes. So there's a lot of powerful and dangerous things that are inside these tubes that you don't want to leak out. That's what stratified cuboidal cells do. They help you to form a protective tube. And that's artist rendition there. We also have a stratified columnar. This consists of several layers of column-shaped cells, and it functions in protection and secretion. It's found also in the urethra, especially the male urethra, and the vas deferens. This is the tube that carries sperm cells from the epididymis to the outside of, to the urethra. The pharynx of males and females. Pharynx is the back of your throat. The anus and the conjunctiva of the eye, which is the clear membrane of the part of the eye, the white part of the eye. Over the surface of the white part of the eye, you have the conjunctiva, which is a protective layer of stratified squamous epithelium. And that's just artist rendition. So what structure is being pointed to in this image? The structure right there. And if you said basement membrane, you would be correct. Well, to kind of wrap up epithelial tissues, let's talk about glandular epithelium. Glandular epithelium is composed of cells that function to secrete substances. So these help to make glands up. And we do have exocrine glands and endocrine glands. So based on where the products are being secreted, we have exo to the outside and endo to the inside. So exocrine glands secrete products onto the external or internal surfaces. Endocrine glands are going to secrete their products into blood or the body fluid. And there's going to be 3 types of exocrine glands: merocrine glands, apocrine glands, and holocrine glands. Merocrine glands secrete watery substances. Apocrine glands secrete portions of cells. Holocrine glands secrete whole cells in their secretion. Now, that's kind of hard to remember, so you want to get some kind of mnemonic or something that might help you to, some kind of learning device that will help you to learn that. So here we have a merocrine gland. An example would be the pancreas, and it's going to be secreting watery-like substances. Typically digestive enzymes is what those things are secreting. Apocrine glands, an example would be the mammary glands. Portions of cells are breaking off. Okay, so when you drink milk, you're drinking portions of cells that came from cows. Holocrine gland, an example would be the sebaceous or oil gland of the skin. And these are going to be whole cells that are breaking off, that are forming the product. So the oily secretions that lubricate your hair follicles and lubricate your skin, those are whole chunks of cells that are full of fatty material. Okay, so I think this is probably a good stopping point. We'll stop here for this portion of the lecture, and in part 2 of histology, we'll do the connective tissues. So until then, I will see you then.